Lots of open presentations here. Okay, thanks, Mark. Hi, my name is Iddo Friedberg. I'm one of the organizers of this meeting, so if you like it, great. If you don't, come to me and let me know why. Hopefully it seemed like a pretty full meeting, so that's great, and we heard a lot of great talks. I'm also responsible, along with a lot of other people, including Predrag Vojac and Casey Green and many others, to the Critical Assessment of Function Annotation Challenge. And nothing is moving here. Ah, that's it. CAFA, or the Critical Assessment Annotation Challenge, or CAFA, is basically a time-set challenge in which we start somewhere around September, and we release a set of protein targets, of which some functions are known, some aren't. And we asked people to annotate them using Go terms so that everything is standardized for about 4 months. And around early February, the year after, we close the submissions and we wait. And some of the proteins that had no annotations gain annotations at that time because, papers get published, and more what happens really is that biocurators insert the data into the data sets. We use SwissProt mostly. And around June, we have a very small fraction of those proteins whose functions were not who were not annotated, experimentally annotated. Around June, they become annotated. So it's a very small fraction, it's like 3%. But 3% of 100,000 proteins is still 3,000 proteins. So it's a sizable benchmark by which you can test your methods. And all the methods here that we talked about, quite a few of them were actually participants in CAFA, or hopefully will be soon. So the idea is that we test these methods and say, okay, how well did you predict based on this benchmark that we generated? And the nice thing about this benchmark, it's so blinded that we even don't know what's going to happen. Although we heard a fantastic talk by Megan Crow where she used cold reading to predict, and that's actually a pretty decent method. Jesse Gillis's group used that a couple of times in CAFA, and they did interestingly well. But that's something for another talk. So anyhow, the preliminary benchmark collection is in June. We have another one around November or December, and then we assess the methods and we publish a paper. We're into our third CAFA challenge now, and the paper is now in revisions. We're pretty sure it will be accepted soon in genome biology. These are the performance of the methods historically in CAFA. Green is... the green bars are CAFA 3, the third CAFA challenge. The yellow bars are CAFA 2, and we mix the top 12 methods according to their performance. Fmax is a kind of an F1, is the maximum F1 precision recall of each method. And the blue and red are the baseline methods that we're going against. Red is basically the priors. We call it the naive methods. So what you expect just by whatever kind of bias you have in your databases. And BLAST is BLAST, okay? So the kind of standard against which we perform. And we can see that in CAFA 2 and CAFA 3, performances were not that much different, except for maybe one method from the Jew lab at Fudan University who did fairly well in the biological process ontology. All these are the ontologies of the different ontologies of the Go terms, and biological process is the ontology that describes the process a protein participates in. Molecular function is the actual what it does, enzymatic function and so on. Cellular component is where it's at. And those are basically the way things are going right now, which shows us that contrary to the improvement between CAFA 1, note that CAFA 1 methods are not here at all because they've been much improved in CAFA 2. Between CAFA 2 and CAFA 3, we had some incremental improvement, especially in molecular function, but not as dramatically as between the first challenge and the second challenge. So that is interesting. Another interesting thing is to see the bias in cellular component. You see that the naive method scores very high there. That particular data set is fairly highly biased, and we're still scratching our heads over how do we deal with that. Another new thing that came into CAFA, we called it CAFA Pi because it was like an addendum to CAFA 3, but not quite CAFA 4, so CAFA 3.14, Pi, haha. And here what we did some high throughput screenings, not we, our experimental collaborators. So that was GeoLab at Dartmouth and Deb Hogan's lab, same. And they basically, this is a nice screen that I can talk about more in my poster. It basically checks long-term memory in Drosophila. So long-term memory in a fly is, of course, something like 24 hours, but you can actually test that, assay for that. And we took CAFA predictions for what long-term memory would be in Drosophila, and we gave it to Gio's lab, and Balint, his student, did the assays, and a surprisingly interestingly number of those genes were actually discovered to have something to do with long-term memory. So here we have computation driving experiments, and it's great, and this paper was published, and you can read it. It's a really nice paper that's published in Gene Genomes and Genetics. I'll skip this because I don't have time. One interesting thing that we found out was that if we compare the top CAFA Pi predictors in light blue to the baseline predictors in dark blue, we see that, yeah, they predict fairly better than BLAST, but look at the interesting thing. There are two types of BLAST here, BLAST Comp and BLAST. BLAST Comp adds data that is computationally annotated that's supposedly kind of not very good, but it actually improves the predictions. Whereas BLAST, only on experimental data, is not as great. So that's actually an interesting thing. More data that came out of Deb Hogan's lab had to do with biofilm formation in Pseudomonas. And with biofilm formation in Pseudomonas, what we can see is that the top CAFA methods, their prediction is, only so 0.58, 0.57, the Fmax. But if we take simple gene expression data and just use that, correlation using gene expression, it predicts much better. So gene expression does a lot better, and that was done in Casey Green's lab. So Casey Green actually won CAFA Pi here. He was very happy about, having like 5 minutes of work and doing better than everybody else. Of course, for that, you actually need gene expression data. So that tells us that we can improve quite a bit more by incorporating things that are not sequence data only, but take a lot of metadata. And that actually sort of also tells us, hey, we could use a lot more RNA-seq experiments, and people have talked about that here. So that's interesting. And if you didn't get what I was talking about right now, my poster is up there, and I'll be there. And time to discuss CAFA 4. CAFA runs every 3 years, and this September we're supposed to do it. We're a little bit antsy about whether we're going to do this or not, and how we're going to do it, because unfortunately our NSF grant was not renewed yet. It was rejected actually. We are asking for a renewal. So we'll have to do it on a low-string budget. We had this great grant where we wanted to do a lot of zebrafish experiments and various hackathons and open source stuff and whatnots. Unfortunately, they didn't quite like it. We're resubmitting. We're pretty high there, but not quite high enough. So we will probably run CAFA 4 starting September. Those of you who are CAFA participants will get notified on the list. Those who are not and would like to learn more, please contact me, and I'll be happy to talk to you about CAFA. We're very easy to find. Google CAFA protein function prediction, whatever, you'll find us, and you can just email me directly or one of the CAFA organizers. So CAFA 4 will happen. What exactly will happen in CAFA 4? That's really up to us because we're kind of under budgeted right now, up in the air. So thank you very much for listening to me. I'd like to thank the people who actually did the hard work in this, who are our students. So Nahi Zhou in my lab, Yuxiang Jiang in Pedja Rodivoric's lab, Balint in Gio's lab, and Kim Lewis in Deb Hogan's lab, who did all the really hard work to generate this. Poster C12 is up. You're welcome to come and talk to me more about this. We have the CAFA website where you can go to. The CAFA 3 is up in a preprint in bioRxiv, and I'm done. Thank you very much.